students, and welcome to our channel Science eSolution. This is Manju Sharma taking your bio classes, and in today's class we'll be covering a very important topic, glycolysis. Before jumping into the details of the topic, kindly hit the subscribe button because you'll be benefited a lot. If you are a bio student, you'll love the video lectures and also the PowerPoint presentations. At least it would be much beneficial for you. So let's go into the details of the video. So from where the name glycolysis has been originated? It's from the Greek word where glykos means sugar and lysis means splitting. So it means splitting of sugar. It is also known as EMP pathway in the name of the German scientists who discovered this pathway. They were G. Empton, O. Meyerhoff and J. Parnas. Students, you can also take down the notes wherever you think the points are important, or you can take screenshots as well. So definition, what is glycolysis? During the process of respiration, glucose is converted into pyruvic acid through a series of enzyme-catalyzed reactions. Glycolysis is actually a series of reactions. There are 10 reactions in the series. And this series of reactions is known as glycolysis and takes place in the cytoplasm of the cell. Okay. Glycolysis is the first stage in the breakdown of glucose and is common to all organisms. When we say common to all organisms, it means that it is common to both aerobic or anaerobic, unicellular, multicellular, prokaryotic or eukaryotic. So any type of organism, it needs glucose for producing energy, and glucose is produced by breakdown of it, and other steps may not be common in all organisms, but this first step, glycolysis, is for all organisms. So let us see the different steps of glycolysis. Here you can see in the first step, glucose is phosphorylated to glucose 6-phosphate. With the help of enzyme glucokinase, we will also be looking the details of these steps in the same video. So stay tuned, subscribe onto our channel and kindly hit the like button. Okay, in this step, one ATP molecule is utilized and it is consumed and it forms ATP molecule. Okay, and glucose 6-phosphate in the next step, it is converted into its isomer fructose 6-phosphate with the help of the enzyme phosphohexose isomerase. In the next step, fructose 6-phosphate converts into fructose 1,6-bisphosphate. Again, 1 ATP is consumed in these reactions. So a total 2 ATP you can see are consumed in the first phase of glycolysis. And then this fructose 6-bisphosphate, it undergoes splitting into 2 3-carbon molecules. One is glyceraldehyde 3-phosphate, which enters into the second phase of glycolysis, and the next one is dihydroxyacetone phosphate, which cannot directly enter into the second phase, but it gets converted into, as you can see, they are reversible. So dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate, and then the glyceraldehyde 3-phosphate enters into the reaction. So second phase of reaction of this 3-carbon molecule glyceraldehyde 3-phosphate, you can assume that this reaction takes place 2 times. Okay, so any molecule produced or used up, we will take it multiplied by 2. So glyceraldehyde 3-phosphate in the next step gets converted into 1,3-bisphosphoglycerate, and in this step 2 molecules. Okay, because 2 times this reaction is going to take place, so 2 molecules of NAD will be converted into 2 molecules of NADH. This is a dehydrogenation reaction. In the next step, 1,3-bisphosphoglycerate converts into 3-phosphoglycerate and 2 molecules of ATP are formed. Okay, because this reaction takes 2 times, so every molecule formed or consumed, we will multiply it with 2. I am repeatedly telling this, so please don't forget to calculate this as 2 molecules while you do the reactions of glycolysis. So in the next step, 3-phosphoglycerate converts into 2-phosphoglycerate and then in the next step to phosphoenol pyruvate with the help of enzyme enolase, and finally pyruvic acid is produced with the help of enzyme pyruvate kinase, and here 2 molecules of ADP are consumed and 2 molecules of ATP are formed. So overall you can see 2 molecules of ATP are formed here and 2 molecules of ATP are formed in this reaction. So a total of 4 ATP molecules are formed and 2 ATP molecules are consumed in this first phase of glycolysis. So total 4 molecules are formed and 2 molecules are consumed. So a net gain of 2 molecules of ATP. So let's see these reactions into details. Students, I'm repeatedly saying, wherever you think the points are important, kindly screenshot them or take down the notes. First step, it is called as the phosphorylation of sugar. That is first phosphorylation, where glucose is phosphorylated to glucose 6-phosphate and 1 ATP is consumed in the reaction. Reaction takes place with the help of enzyme hexokinase, or more particularly glucokinase, and magnesium enzyme and magnesium ions are also needed. So glucose plus ATP, they form glucose 6-phosphate and ATP is consumed and is formed ADP. In the next step, subsequently isomerization of glucose 6-phosphate to fructose 6-phosphate takes place. So glucose 6-phosphate with the help of enzyme phosphohexose isomerase and magnesium ions converts into fructose 6-phosphate. The next point is phosphorylation of fructose 6-phosphate, which is also the second phosphorylation. So after fructose 6-phosphate is phosphorylated, it forms fructose 1,6-bisphosphate. And the enzyme required for this process is phosphofructokinase. Okay, 1 ATP is also consumed in this process. So fructose 6-phosphate plus ATP in the presence of the enzyme phosphofructokinase and magnesium ions, they form fructose 1,6-bisphosphate and ADP is released. Students, we are discussing the details of glycolysis reactions. Then in the next and very important step is splitting. This fructose 1,6-bisphosphate, it splits into 2 molecules. This is a 6-carbon molecule. Okay, this is a 6-carbon molecule which converts into 2 3-carbon molecules. This is phosphoglyceraldehyde. This is a 3-carbon molecule, and dihydroxyacetone phosphate. This is also a 3-carbon molecule. Okay, so this is a very important step, that is splitting, in which a 6-carbon molecule fructose 1,6-bisphosphate with the help of enzyme aldolase converts into 2 3-carbon molecules. One is phosphoglyceraldehyde and the another one is dihydroxyacetone phosphate. And both of these are interconvertible. That means phosphoglyceraldehyde can be converted into dihydroxyacetone phosphate, and dihydroxyacetone phosphate could be converted into phosphoglyceraldehyde. Actually, dihydroxyacetone phosphate only converts into phosphoglyceraldehyde and enters into the second phase of glycolysis. The next step is called as oxidative dehydrogenation, in which 3-phosphoglyceraldehyde is oxidized to a carboxylic acid, which is 3-phospho-1,3-bisphosphoglycerate. You can see here, and NAD is reduced to NADH. The enzyme used for this reaction is glyceraldehyde phosphate dehydrogenase. The next step is formation of ATP. ATP molecule is formed. Actually, there is release of 2 ATP molecules because this reaction occurs twice. One when glyceraldehyde phosphate enters the reaction, and the another one when dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate and enters the reaction. Okay, so enzyme phosphoglycerate kinase, it acts on 1,3-bisphosphoglycerate, okay, and forms 3-phosphoglycerate with the help of, and also ATP is released in the process. Okay, come to the next point where their isomerization occurs. 3-phosphoglycerate converts into 2-phosphoglyceromutase. 3-phosphoglycerate gets converted into 2-phosphoglycerate. It is written wrongly, so it gets converted into 2-phosphoglycerate. While making the presentation, I think there is some mistake. So 3-phosphoglycerate converted into 2-phosphoglycerate with the help of the enzyme phosphoglyceromutase. Okay. Then this is the main reaction, and the next step, there is dehydration. This is actually the last step in which 2-phosphoglycerate with the help of enzyme pyruvate kinase converts into 2-phosphoenol pyruvate, and water is released. That is why it is dehydration reaction. And in the last step, 2-phosphoenol pyruvate with the help of 2 ADP molecules gets converted finally into pyruvic acid. 2 molecules of pyruvic acid are formed and 2 ATP molecules are produced by this reaction. So you can see in the overall chart, 2 NADH and 2 ATP molecules are produced at the end of glycolysis, and also 2 pyruvic acids are produced at the end of glycolysis. So students, hope you have understood the whole concept of glycolysis. You can go through one detailed reaction in which all the 10-step series of reactions are given, and also these are the detailed steps are presented in different slides. I hope after all these presentations you have understood the concept. Kindly hit on the like button if you have understood the concept, and subscribe onto our channel. Stay tuned so that you get benefits from our next subsequent lectures. Thank you for watching.